I think science is inherently a very creative process. A lot of people perhaps who don't work in science don't think of it that way because of their experience in school. But research science is, I think, intrinsically creative, because essentially what you're doing is having to imagine something beyond what's already known.
Hello and welcome to Everyone Is. I am your host, Jennifer Coronado. The intent of this show is to engage with all types of people and build an understanding that anyone who has any kind of success has achieved that success because they are a creative thinker. So whether you are an artist, or a cook, or an award-winning journalist, everyone has something to contribute to the human conversation. And now, as they say, on with the show.
I learned about neurologist Dr. Joanne Jones through a colleague, and I was so impressed to hear about someone who's made it their life's work to work towards eradicating a disease like multiple sclerosis. I feel pretty honored to have her join us here today. So welcome, Dr. Jones, to Everyone Is.
Thank you very much. Thanks for inviting me.
My film nerd in me also wanted the opportunity to say, welcome, Dr. Jones. So,
yeah, Indiana, I can be called from now onwards. That's fine.
That's right. Where are you from and where did you grow up?
I'm from a city called Wolverhampton in England. So it's a city in the centre of England in an area called the Black Country, fairly industrial area of the UK originally, a fairly deprived area in many respects. But yeah, I grew up in the Midlands, but now living in Cambridge.
As a kid, were you into science? What were the things that you were really interested in when you were little?
Yeah, I mean, I've always been interested in science, trying to work out how things work. That's very much how my brain processes things. But to be honest, at school, I liked most subjects and I was pretty good at most subjects. So I was fairly undifferentiated, really, I'd say, until probably my early to mid-teens, where it became a bit clearer to me that it was science, that I was the direction I was going in. And I ended up realising that I could do medical research. That was the key for me. So I remember actually seeing an old black and white film about Marie Curie. And that I ended up coming away from the film thinking, gosh, I want to do some sort of research, make a difference. And that was actually my first realisation that you could do research. But at that point, I didn't really realise you could do it as a doctor. And in conversation with one of my teachers, that she pointed out that actually science and medicine can go hand in hand. You don't have to choose between the two. You can be a researcher and a medic. So that's how I decided to do that.
When you're thinking about, as a kid, I can make a difference, what did that mean to you?
Good question. I think it was around health. It was always around health rather than, it was that aspect of science, realising that there were various untreated diseases. I read a lot. I read a lot of scientific books. I read a lot of scientific journals, things like The New Scientist when I was a teenager. And hearing about advances in science and advances in medicine and how people were making a difference, that was really what interested me. I wanted to do something useful with the science, not just learn science for science sake. So it was always around health. But as I say, originally I thought that you had to be either a scientist or a doctor. And I didn't really have any idea that you could do both in parallel until I was probably 16, 17, really, when I had this quite pivotal conversation with one of my teachers, when she pointed out that actually you can do both together.
Yeah, it's really interesting. I know when I was a kid, I was very interested in political science. And I thought, the job is political scientist. I didn't realize you could apply that job to so many areas. And it is those revelations that are very interesting when you're a kid evolving into an adult.
It's quite difficult because you have quite a narrow experience of what jobs are. So it's very easy to imagine what a doctor is from visiting your general practitioner or seeing doctors in medical dramas on TV or in films. And those doctors were never doing research. They were always looking after patients, often in a very dramatic way. I think until you actually start to speak to people and get advice from people who work in these fields, that you actually realise the breadth of what you can do with both science and medicine.
You attended Cambridge for your PhD in neurology, but after you attended the University of Bristol to become a doctor. So what did that path look like? Because were you a practicing physician before you went to get your PhD?
Yeah, so I did my undergraduate degree in Bristol. So I did my medical degree there. I also did a Bachelor of Science degree in Bristol. So it's something called intercalating. So the medical degree in the UK, and I think in quite a lot of countries, is tends to be split in half. So there tends to be a period that is very book-based learning, and then a period that is very kind of experiential, on the wards type learning. So the first is a 2-year part of your degree, the second a 3-year. And between the two, if you were doing well in the book side of things, you could do a Bachelor of Science degree in one of your medical specialities, as it were. And I did a Bachelor of Science degree in microbiology, but very heavily immunology degree. I did that in a year. I did my medical degree and a Bachelor of Science degree. So I left medical school knowing that I wanted to do some sort of immunology research because of this year. But yes, I went on the wards and did all of my postgraduate medical training. So, worked in accident and emergency, so ER equivalent, and did all my medicine rotations, so cardiology, respiratory, elderly care medicine, etc. And then I realised that I wanted to do neurology. And that really was because when we would admit patients on call overnight, the patients that tended to be the most interesting to me were the ones with neurological disorders. They were a little bit more difficult to work out what was going on. I found myself wanting to know what happened to those patients after they'd been admitted. So I realised that neurology was the path I wanted to take. And there's a whole subspeciality of neurology called neuroimmunology, where the immune system is interacting with and attacking often the brain or the spinal cord or the peripheral nerves. So I decided that was the research that I wanted to do. So I did my PhD, as you say, in Cambridge, but I was still training as a neurologist. So I took time out of my training program, so just over 3 years out to do my PhD, but I was still doing clinics occasionally, still doing on-calls occasionally. And then after my PhD, went off and completed my neurology training to become a consultant neurologist. So it's very much about doing the two in parallel.
What is a consultant neurologist? What does that entail?
So it's the most senior doctor. It's the doctor who takes responsibility for the care of an individual patient and has a team of doctors working for them. So I'm a consultant neurologist with a subspeciality interest in multiple sclerosis. So I look after patients with MS, and I'll have more junior doctors working with me, looking after the patients.
So you're managing teams then?
Because I do my academic work, I work about 40% of my time doing clinical work and 60% of my time in the lab. I am involved in managing a team, but I'm not the main manager of the clinical team. There are pure NHS consultants who manage the team, but I'm one of the consultants who manage the MS team in Cambridge.
Cambridge was founded in like the 1200s, and the medical school was founded in the 1500s. There was a reemergence of it in the 1970s, and it's one of the most prestigious, if not the most prestigious medical schools in the world. Is there a weight to that as a physician, or does it feel like old hat to you at this point?
Cambridge is one of the oldest universities in the world and is one of the most prestigious universities. A lot of competition between Oxford and Cambridge in the UK. So I trained in Bristol, which is in the southwest of England, and worked around the southwest. And I first came to Cambridge. I think I was quite anxious, would I fit in? I was this person who'd grown up in the West Midlands, which is quite a deprived area. I'd gone to a very good university. Bristol's is one of the top universities in the UK. But I was quite anxious about what Cambridge would be. Would I feel somewhat different from the average person in Cambridge? And that hasn't been my experience at all. The department I work in is incredibly friendly, incredibly supportive. I mean, it is a very competitive environment, Cambridge. So there is the pressure because of how competitive it is to get here and to remain here, but it is a supportive environment as well. And that anxiety that I was somehow not Oxbridge enough for Cambridge, which is the term we use for kind of Oxford and Cambridge together, Oxbridge, that the anxiety that I wasn't Oxbridge enough because my background wasn't, I come from an academic background, I didn't come from a wealthy background. That really dissipated when I was here. But I think that's largely because of my PhD supervisor. So I was lucky to be supervised by somebody called Alistair Coles, who was and continues to be incredibly supportive, and the department that I came into in Cambridge. It's important for me now to go out and to tell people not to be put off applying to places like Cambridge because feeling that you somehow shouldn't fit in here, because actually that isn't the case. And it certainly is a big part of Cambridge trying to open up its recruitment so that people from all sorts of backgrounds, apply. They want to take the best, but irrelevant of background.
Yeah. Sometimes it's interesting. Sometimes people don't understand that you have to think you have an opportunity to be the best in order to apply for something that's defined as the best. Like that self-doubt that coming from a background where you don't feel like you're quite right for this world can actually eliminate really talented people from pools that are really great thinkers and scientific problem solvers. I want to go back to the West Midlands a little bit. What did your parents do when you were growing up?
My parents aren't academics at all. My father worked in a factory, but as sort of middle management, really. So he was a work study engineer, but he'd worked his way up from being somebody who worked on the shop floor and then had become trained, and his seniority increased during his working life rather than going in with a degree. And my mom was a telephonist, so used to work actually in a hospital. It's called an eye infirmary, so specialising in care of eyes and eye emergencies, but on the answering the telephone for the eye hospital. So not at all an academic background. My parents are both, very bright, very capable people, but, came from very working class backgrounds themselves. And I think, they're both in their 80s. And certainly at that time in the UK, it would have been difficult for somebody of their background to have gone to university. Not impossible, but very difficult.
What did they think about your role as a doctor?
Very proud. When I first started to do medical research and sort of moved away from being a full-time clinician, it probably took them a little while to get their head around what that meant, because to them, being a doctor and working in a hospital, being a consultant seemed, perhaps, you'd made it. So to suddenly then shift sideways somewhat to then start to train in parallel as a scientist, I think they found that a little bit strange. Why was I doing that? Why was I taking time out? But they were incredibly supportive. My parents have always been very supportive, not at all pushy, but always supportive. But now I think they understand why I like doing what I do, and they are incredibly proud of me. And, I know that they delight when I tell them about the sorts of things that we're doing, and they are interested in the research, and I try and explain it to them. So, yeah, I'm very proud.
That's great. You talked about, how you saw that, in essence, neurological disorders when you were working in the hospital, and that became an area that was very interesting to you. And I know that multiple sclerosis is an area that you focus. But I wonder if you could talk a little bit about human autoimmunity as it relates to that. And what is autoimmunity, just on a base level, if you were just explaining it to a Luddite like myself?
So we use the term autoimmunity when we're meaning that the patient's own immune system is attacking a bit of their own body. So our immune systems, and by that we mean really our white blood cells that are in circulation and that are trying to mainly fight infections. It's their primary job. When they're developing, your immune system gets trained in the thymus, which is a gland in the body, to not attack itself. So it gets shown self and told not to attack it. It goes through a sort of training system in the thymus. What happens in an autoimmune condition is, for some reason, the immune system, that process hasn't been perfect, and the immune system can then attack parts of the body. So a good example of an autoimmune condition is something like rheumatoid arthritis, where, most people have seen people with inflamed or hot, swollen joints. In multiple sclerosis, the same sort of thing is happening, but it's happening in the brain and the spinal cord. So patients are having episodes of inflammation. So episodes where their immune system, their immune cells are trafficking into their brain and their spinal cord, causing that inflammation in a similar way to you'd see in a joint, but happening hidden away in the brain and the spinal cord and causing damage. And then typically what happens in MS is people will have a wave of inflammation, and then that inflammation will settle and they'll have a remission. But after a period of time, and that's highly variable, but it might be a few months or a year or so, they'll have another attack and they'll have another episode of damage. They might make a good recovery from the attacks, but often what happens is over time the damage builds up and patients start to acquire disability, which is why it's so important to stop those attacks from happening in the first case.
Medicine is built on layers and layers and layers of previous research, right? So as you're pushing research forward, you have to really absorb all the research that has happened prior to that. So, number one, you're not going down the same path and ending in a dead end. And number two, you're actually pushing the medical agenda forward where you can actually look towards solutions. So, how did you, as an individual, when you were learning, absorb what you needed to know about this specific disease in order that you can start to find different paths forward to combating it?
That's a good question. And to be honest, I think that the vast amount of information and papers that are published around all subjects in science, it is actually very difficult to keep on top of the literature. I mean, it's a job in itself to continue to, if I read every single paper that was written on multiple sclerosis, I would literally spend my entire life just reading papers and wouldn't have time to see patients or, actually do any research. So it is a struggle to keep on top of things. But I guess in terms of how I started to think about what I wanted to research, it came from my PhD. I knew I wanted to study, because of my doing my Bachelor of Science degree in immunology, it was in microbiology, but essentially what I studied was predominantly genetics and immunology. And it was the immunology that I found really fascinating. How if you were to have a kidney transplant, you'd have to be on drugs to prevent yourself from rejecting the kidney. But how is it we can eat food and our stomachs and gut not reject the food that we're eating, for example? I remember that being something that I hadn't ever thought of. And then suddenly having this presented to me during a lecture, I was thinking, gosh, this is incredibly fascinating. How complex the immune system is. How can a woman become pregnant and carry a baby that is genetically distinct from herself and not reject the baby? So, all of these questions suddenly, I hadn't really thought about them, and having them presented in the course was, a real eye opener. So I knew I wanted to do immunology. I knew I wanted to do neurology, and MS, I guess, is the classic neuroinflammatory disorder. So I was looking for a research opportunity and a supervisor who was working in that field. I came to Cambridge because Alistair Coles, who was my PhD supervisor, and also Alistair Comston, who was the head of department and also another supervisor of mine, they were developing a drug called alemtuzumab, which is now licensed under the name Lemtrada for multiple sclerosis. Joining their research team meant that I could work on a clinical trial, which I wanted to do because I'd not done that before, and still get to see patients. But also it gave me the opportunity to have access to samples from patients, so I could start to try and dig into fundamental immune mechanisms. And what I looked at during my PhD was this drug, which is a fantastic drug. It's still used as a treatment for multiple sclerosis. What it does is it switches off patients' multiple sclerosis very effectively, but half of all patients, after they've had the drug, actually develop another autoimmune condition, predominantly affecting their thyroid gland. So I was working on how can this drug switch off one autoimmune condition and switch off another? What's it doing to the immune system? And that was essentially what my PhD was on. And that got me interested in fundamentally, how does the immune system work? How does it regulate itself? How does it prevent autoimmunity? And it was really from that I've become interested not just in multiple sclerosis, but more generally how the immune system regulates. So my lab, we still very much work on multiple sclerosis, but we also work more fundamentally on a type of immune cell called a regulatory cell. So these are the cells that do the act of regulating and suppressing the rest of the immune system. So my lab works on how do those cells achieve that and why do they go wrong in diseases like MS. And we also work in other neurological disorders. So we now know that conditions such as Parkinson's disease and Alzheimer's disease and other neurodegenerative diseases that were previously thought not to have anything to do with the immune system, we now know that the immune system also plays a role in those diseases, probably as a secondary event. Something happens in the brain, there's some aggregation of protein that shouldn't be there, but then the immune system goes in and causes secondary damage. So we also work on those disorders, and we try and work out what makes up the human immune system.
And it's because there's things that you study, like the regulatory cells, that are part of that research.
Yeah, so regulatory cells are part of our immune system. They're really key. They are important in preventing the immune system from inappropriately getting activated and attacking self. they're also really important in switching off immune responses that have occurred appropriately. So in response to infection, for example, you need to clear the infection and then switch off the immune response. And regulatory cells are involved in all of that. So we try and understand what's the nature of regulatory cells in humans. So there are lots of different sorts of cells that can play that role. And we've identified a cell that is somewhat different from the cell that people mostly talk about. So cells express lots of different proteins. We've identified a regulatory cell that seems to be very important in human tissue. So some of the work that we've been doing is obtaining tissue from individuals who are on the organ donor register. So individuals who've died and very kindly gifted their organs for transplant, and their families have given permission for researchers to take additional small samples of tissue from around the body. We've been looking at that tissue and been trying to understand what's the composition of the immune system in the different tissues across the body, and also what about these regulatory cells? Do they sit in these tissues, and what role might they play in the tissue? So that's what my lab's been doing quite a lot of over the last few years. And we're now taking that research back into disease. So we now understand better what the immune system of the gut looks like versus the immune system of the lung versus the immune system of the kidney versus the immune system of the blood. We now understand the immune system better, and we're now trying to understand how does it become perturbed in disease, and these regulatory cells that are sitting in tissue, how are they involved in particular diseases? So we've come full circle back to saying, what changes do we see in multiple sclerosis?
It's interesting. The human body has so much because there's so many specialists, and sometimes you wonder, oh, there's a heart specialist, there's a immunologist, and you're like, well, why do we need all these specialties? But the human body is a machine with so many parts that and so many interconnected things. Specialties are important in order to understand how it works as a whole.
It is absolutely fascinating how the body as a whole and the immune system works. And it is so intricate and so detailed and so designed to work within quite tight parameters. You know, our blood pressure should be a certain range, our heart rate should be a certain range, you know, our temperature should be a certain range, our immune system should work in a certain way. And there's all of these amazing control mechanisms that the body has to deal with all of these things. And then it has to be able to cope when it's perturbed in some way. So an infection or an injury, it has to be able to respond to that, heal tissue, respond to infections, prevent tissue damage from infections, try and prevent cancers from being, generated. And the immune system plays a very big role in clearing cancers. So absolutely, both on a clinical level, you need specialists who understand this sort of vast complexity of the human body and the diseases that can happen to various bits of the body. And also from the research side, you need people who are looking at different aspects and different systems. But I think what's important is that all those people do also talk, because often there's similarities and connections and things that we can learn from each other. And obviously there are some disorders that, and especially the immune disorders actually, that from the clinical side, they cross specialities. So certain diseases will not just cause inflammation in the nervous system, but they might also affect the kidneys at the same time, or they might affect the skin at the same time, or gut. So there are disorders that cross multiple systems.
My mother had multiple myeloma. And what you realize when people have a cancer of that nature is that so many things can go off. Like suddenly, you're being treated for the cancer, but now your kidneys are being impacted, or your heart is having, you build up amyloidosis in there's so many balances that your body is constantly working on that learning about that has to feel like so challenging. That brings me to the question, are there ever any aha moments that you've had in your research where you've discovered something that was new to you or just new in general that you went, oh, now I understand how that works, or now I understand how to approach that? Have you had any of those moments?
I think we've had I've had, and my team have had lots of small aha moments where something suddenly slots into place. So for example, with the regulatory cells that I was telling you about, they're called CD8 Tregs. But with that particular cell type, we looked at what genes the cells were expressing in the blood. So what genes and what proteins were these cells expressing? And what we found is that they were expressing molecules that would suggest they might traffic into tissue. Because we'd been wondering, why do you need another type of regulatory cell? There's the types that people had already been working on. So why on earth do you need this one as well? What's the difference? And then we looked at the way these cells were expressing these molecules and thought, I wonder if they're in tissue. And that was a little bit of a aha moment, because we were already looking at what the immune system looks like across the human body in all of these different tissues. We could go and look. We had the access to this really precious material from these organ donors through this amazing resource in Cambridge called the Cambridge Biorepository for Translational Medicine. We had this amazing opportunity to actually look, and we saw that they were there, and they were there in increased numbers than we were expecting. And again, that was a bit of a aha moment that, gosh, this is really something important, and we're now trying to understand these cells in more detail. But I think a lot of science is very incremental. So exactly as you were saying before, it's often somebody will publish something or a technique or something they found in their disorder that subtly shifts the way you think about what you're doing, and you wonder whether something similar could be happening or something related could be happening in what you're studying. So you have little moments. I think these kind of big Archimedes moments where you sit in the bath and suddenly realise water displacement is a very rare event, although I'm sure occasionally they do happen. But I think it's smaller than that. But yeah, I think you do occasionally get the ahas in medicine and science, which is a very special moment when it happens.
Getting a little bit back into your roles, you have two designated roles at Cambridge. It's associate professor, and the second is the clinical lecturer, which we talked a little bit about that. But tell me, is your role as associate professor work at Cambridge?
Yeah, so the associate professor role, that title in Cambridge reflects me running my research group. So that's me running my team and my research. I run a team of people, so a wet laboratory, so we're doing wet lab work, and I have a fantastic group of people that I work with. I have 5 postdoctoral research scientists who work for me, and I currently look after 5 PhD students and 1 MPhil student, and I also have a fantastic lab manager. We're a very happy group, I think, or hope. So now I don't really do much wet lab work myself.
I'm going to stop you 1 second and just say wet lab work. Can you define that a little bit for the general audience?
So wet lab work meaning where you're actually at the research bench, pipetting liquids, dealing with tissues, dealing with biological samples. Yeah, we use that term wet lab work to distinguish from people who do, for example, research, but might be handling big data sets or looking at imaging or something like that. Doesn't require that handling of fluids. And
it's literally wet lab work.
It's literally wet. Yeah, exactly. I now really don't do any bench work. If I do ever do any bench work, it causes quite a bit of amusement in the group that I'm handling a pipette, because it's a very rare event. So most of my job is coming up with new ideas, writing grant proposals, trying to get research funded, which is obviously quite a stressful part of my job, ensuring that people have their salaries. So you function as a small business when you run a research group, because you're having to bring in funds from, a whole variety of different funding agencies to fund your research and to fund people's salaries. Overviewing all of the projects, so ensuring that the projects are on track, reviewing the data, discussing the data with either the senior scientists in the group or the students in the group, and talking about the direction or the things that we should be looking at. That's the fun bit. That's much more fun than writing grants. Actually looking at the data with the people in the group is fantastic. And, problem shooting and doing lots of other things that are much more boring. So dealing with contracts, checking that samples can be sent around the country, and dealing with the sort of paperwork side of doing research. But the fun side is the supervision of the projects and the training of the PhD students and the MPhil students. I have to say that's a real joy of my job, because you're working with and training and seeing the development of 20-year-olds. And I think that keeps you young, having those people in the group. It's really lovely to work with young people. And I walk into my lab and people will be busy working, but you'll hear lots of laughter and people chatting. And that's a real joy, I think, of working in a group.
There's an experience that comes with when you're getting older and a wisdom, but you need that complemented with the energy of people who don't know that they're boundaries. Because that's how boundaries get broken and that's how new ideas come up, is that energy of, hey, we can do this. So I can imagine that's inspiring.
It is. And as you say, I think it's the sort of energy and enthusiasm and the excitement over the data, and it stops you feeling a bit worn down by some of the other, aspects of the job, the paperwork, the admin, the contracts, the money side. Sitting with people who are genuinely excited about the data and enthusiastic about their project is really lovely. And I have to say, another joy of being a researcher is the collaborations you build up. So you do get to work with such incredibly talented people, within your own team, but you also, science now is rarely done by single individuals in small groups. A lot of science is big science. And, we work with various groups across Cambridge and beyond. We've got collaborators in the US, in Israel, in Italy, in Sweden that we work very closely with. And it's, inevitably these people are incredibly talented, that it's nearly always a joy to work with people. And, that is a really wonderful part of science.
Yeah. The people part is always the joy and the challenge, right? And I imagine like each new batch of students or PhD candidates that you have had to move along in their careers, there have to be challenges that develop along that side as well. Every person thinks differently. So how do you, as an educator and also a manager of a team, how do you approach your team? What's your process there?
So you're right that people are the joyful aspect of the job, but also obviously also can be the challenging aspect. In terms of the PhD students, you really do see a development of them during the time that they're in the group. I think it's about setting expectations early, what you expect from them and what they can expect from you. So both ways. I try very much in my group to ensure that I am very close to the projects that they're running. You do hear stories of people who are doing PhDs who never see their supervisors, or, they do see them, but, once in a blue moon at a big meeting, and there's very little interaction. That definitely isn't the way I run my group. I have weekly supervisions with my students. I'm also very lucky. So I've set up my lab where I have senior scientists in the lab. I have 5 postdocs, but 3 of the postdocs are very senior. So each of them have got more than 20 years experience of immunology. And that's quite unusual, actually. And I'm really lucky to have those people in my group because it also provides support for the students. So there are people who are there who've got huge amount of lab experience doing that wet lab work that we talked about. So can not only guide them intellectually, but also can guide them in a very practical sense with the assays that they're doing. So I try and set things up so the students are extremely well supported, that they have a nominated postdoctoral scientist who's involved in their project, and I try and meet them very regularly to check that they're understanding their project, but also giving them the freedom to develop their project, because that definitely is something that should be part of your PhD. It shouldn't be that you're coming to do a project that somebody else has designed for you as such, with no freedom. There should be plenty of freedom for you to take your project off in different directions and to actually respond to the data that you're getting. So, to notice things, to see things, and to be responsive to that, and to take things off in different directions, and to do the reading that I can't always do. They should be the experts in their project. They should have read every paper that I can't possibly read about their smaller area. And they should be telling me, this is a fantastic paper and I think I should be doing something similar, or this paper's told me that what I'm doing doesn't need to be done because they've already done it and shown it didn't work, or whatever. So yeah, it's about supporting, but giving them some intellectual kind of creative freedoms whilst keeping their ideas under check and checking what they're doing makes sense. So it is
How
a
do you
balance.
build trust, both for yourself and for them? Because you have a lot to, so how do you build that trust so you can depend on each other?
Yeah, I mean, maybe that would be a better question for my team to find out if I've done it well. But I think being very honest. So, being very straightforward, being very honest about what they're showing me, whether I think what they're showing makes sense or doesn't make sense, being open to suggestions, hearing what they're saying, but giving very genuine and honest feedback. It's difficult. I mean, I think it's about being as straightforward and honest as you can with the people. And when you say you're going to do something, delivering on it so they have that trust in you. And also making sure that they realize that you're wanting their career to develop, because obviously that again is something, particularly as people get towards the end of their PhD, or you're helping people transition to being what's called a mid-career scientist. So someone who themselves is beginning to set up their own lab or wanting to, making sure that you make decisions that, maybe aren't always in your best interest, i.e. I'd like you to stay in my group, for example, but seeing and making it clear that you realize how important it is for them to do things for their career progression and supporting them to do that. And again, I think that's a balance, which I hope I get right. I suspect I probably don't get it right all the time, but I do try.
If you do get it right all the time, then you are the first person in the world to get things right all the time. I know that you're also honorary consultant neurologist at Brooks Hospital. What is your role there?
Yeah, so that's the clinical aspect of my job. So that's me looking after patients with multiple sclerosis. So essentially, that means me seeing patients with multiple sclerosis in clinic, deciding which treatments to start them on, which nowadays there are now lots of very good treatments for the early stage of multiple sclerosis. So we talked a little bit about what MS was a while back, where patients will experience these relapses where they have damage to the brain and the spinal cord, and then we'll go through periods of remission, and they'll continue to acquire damage over time. If that damage is allowed to happen unchecked, patients enter what's called the progressive phase of the disease, where the immune system tends to calm itself down a little bit, but patients just continue to get worse and they acquire more and more disability, typically lose the ability to walk independently, might require sticks or a wheelchair, perhaps cognitive problems with bladder or bowel as a consequence of their MS. So nowadays, we're very fortunate that there's lots of what are called disease-modifying treatments that actually modify the disease. They're not just about symptom control. They're trying to modify the underlying disease process that we can use during that early relapsing remitting phase, with the hope that we can stop people entering the progressive phase. But all of those drugs have potential side effects. They work on the immune system. They suppress the immune system. Some of them deplete part of the immune system. So my job in clinic is to see patients with MS, talk to them about what their MS is and what that might mean for them, to assess them, to look at their scans, but also to decide which of the treatments is right for them. So obviously that's a process with the patient, but to guide the patient through that process as to which of these disease-modifying treatments is the right thing for their MS, but the right thing for them as a whole, as a person. Because some of the drugs, for example, you can't get pregnant on, so we have to take that into consideration. And MS is a disease that typically affects women in sort of 20s and 30s, so often at a period of time when they're thinking about starting a family. So that's my role, really, to help guide which treatment and to do all of the monitoring, safety monitoring, checking that their disease really is in remission. Mm-hmm. But I do that as part of a team.
Do you deal with adolescents as well?
Yeah, I'm trained as an adult doctor. So as a sort of fairly early split as a trainee doctor, you decide whether you're going to do adult medicine or pediatric medicine, and I'm an adult doctor. But I actually have set up fairly recently, over the last sort of year and a half or so, what's called a transition clinic for adolescents who are transitioning between being in the pediatric service to the adult MS service. So this is really looking at children and young adults. So we start thinking about seeing them age about 15, but really they come over to the adult service when they're 16. We decided as a team in Cambridge that it would be useful to have a special clinic to do that, to see these children, to give them the extra support they need, and to help them transition from a service where they're very looked after very carefully, but some of the decisions have been made by their parents, for example, when they were much younger, to now transitioning them to being the people who are responsible for their own disease and for their own, coming to clinic, getting their blood tests done. And as they transition into having to think about things like contraception and all those things that might be difficult conversations to have with their parents present, we decided it was useful to set up a transition clinic. And I'm the adult neurologist who has done that in Cambridge. Fortunately, MS is not that common in children. So, it's not from a workload perspective, it's not a lot of clinics that I do, but I think it is an important service that we offer.
When you say we decided to do that, who's the we in that scenario?
In Addenbrooke's, we work as a team of neurologists. There's a group of us who are MS specialists. I think there's 8 of us. So there's a lot of us who do MS work. We look after, in Addenbrooke's alone, about 1,300 patients on disease-modifying treatments for MS. And there are lots of other patients who are in the progressive phase of the disease who come to various clinics. So there's a mixture of consultants who are full-time clinicians, so full-time NHS consultants who do MS, but also will do general neurology clinics and will be on call for general neurology. And then there are a group of us who are academic doctors who have research parts to our job, who also provide service for the NHS clinic. So it's us as a team. Again, I'm very lucky that the team is a very good team of people, and we have meetings. We have strategy meetings a couple of times a year where we look at the service and say, what could we do better? How can we make the MS service in Cambridge better? How can we provide better care for our patients? And it really came out of one of those meetings that we thought we should be putting in a transition clinic.
As a doctor, how do you provide empathy for your patients, but also protect yourself emotionally? Is that balance even possible?
I think that's shifted as I've done less clinical work, actually. So when I was doing full-time clinical work, when I first qualified as a doctor, and when I first qualified, which was 1998, 99, that sort of time period, in the UK, as a junior doctor, you were still working over 100 hours a week, these awful shifts, not sleeping for 48 hours and being on call the whole time. I think I felt I would at times notice that I was getting kind of compassion fatigue. So it was another patient who'd had a heart attack or another patient who'd come in unwell. I mean, I think I was always kind to the patient, but you learn to see the patient, deal with them efficiently, and then to some extent switch off and move on to the next patient because you were so busy. There were patients that still stick with me that were, particularly difficult cases, or patients that I got to know really well. So I worked in oncology for a while, and I remember one lady very clearly, and her children and her family. And when she passed away, I found that very difficult. But on the whole, when I was doing medicine all the time, I think I built that skin that you have to as a full-time clinician, where you care, you want to do your best, you're as professional as you can be, but at the end of the day, you have to move on to the next patient, or you have to be able to make the difficult decision or have the difficult conversation. I actually find now that I'm doing less clinical work that I'm much more emotionally impacted again by patients, because I'm having less exposure to it. So it's 40% of my time, but only some of that is face to face with the patient. Some of it is doing the other aspects and looking at scans and writing reports, etc. And now when I see patients, unfortunately, it's becoming less and less that we are seeing patients who are very disabled because of these very good drugs. But when I sit in clinic and have somebody who's there, who's very young, who can't walk, who's struggling to be independent, struggling to feed themselves, I do feel quite emotional in clinic, and I actually sometimes find it quite difficult to hide the emotion that I see with the patient. And I think it's because I'm less exposed to it. So it has more of an impact than I think it would on the general population.
Yeah.
Because I'm a little bit more back to being a regular person rather than the sort of clinician that has to deal with stuff day to day.
Well, just to show that you as a human, you always are human. And sometimes you have to, as you said, build up that thick skin to protect yourself. But that humanity is always present there. And it feels like that's the type of reason why you want to go into medicine and to problem solve and to make a difference, as you talked about when you were young, thinking about making a difference. There's lots of new techniques that are coming available to medicine. And one of them is machine learning, the AI impact of making data sets and putting it through computers to find patterns. And I'm wondering how much you're seeing that in your studies and your research work at this point.
It's definitely having an impact already. I mean, even on the clinical side, there are now AI tools, for example, reading MRI scans. So obviously one of the ways that we monitor patients to ensure that we've put them into remission is we monitor them by imaging their brains every so often to check that they're not having any new scars, new lesions. Has the drug worked? And if it hasn't, we can respond to that. And that still is done largely by a neuroradiologist who will look at the scan and will have to very manually go through the images to see whether there's been new lesions. But there are now AI tools that can do this really rapidly, can tell us lesion counts, tell us brain volumes, etc., within a matter of minutes, rather than a much more time-consuming process for the consultant involved. And it's definitely being used in research for handling big data sets, for exploring big data sets. One aspect of the work that we do is we do lots of single cell sequencing. So that's where you look at a population of cells, for example, from within the brain or from within another tissue. And on a single cell level, you look at what genes they're expressing. And this generates a huge amount of data. And you can, from that data, tell what the cell is, but you can also tell what cell state it's in and whether, you can also try and work out whether it's interacting with other cells and things in its environment. And there's lots of computational tools that are available to help you deal with that data, but it's not quite the sort of machine learning that you're talking about. But you do see that coming in, and I think it is going to revolutionize things and move things on at a pace.
As a researcher, a clinician, a doctor, a Joe, what's the most exciting thing you see coming down the road? What's the thing that is there something right now that gets you excited about future developments?
So from the laboratory side, I think it's just the pace of technology. It's both exciting and terrifying because we can, the single cell sequencing that I talked about.
Yeah.
It's very exciting and relatively new, but it has still been around for quite a few years. And now the next stages of that are coming along. So not only can you understand what every single gene a cell is expressing, but you can understand how those cells are regulating what genes are expressing by different techniques that have come along. You can now look at them in a spatial context. So there's now some amazing spatial imaging techniques. So you take brain tissue, for example, and you don't just understand what cells are in there, but you can literally see them at the single cell level in a spatially resolved image. So you can start to understand what those cells are, how they speak to each other, how they are related in a very spatial way to for example, a pathology. So maybe a brain contusion or an MS plaque. You can see what's sitting around that literally in the brain tissue. And the pace of these technologies is just enormous. I mean, it feels like every few months there's some sort of advance. And that's exciting, but it's also challenging because a lot of these technologies are very expensive when they first come out. It's difficult to have the funds to do the very exciting work and also to have the expertise to be able to use all of these different technologies. It's challenging to keep on top of them. But, it is exciting, and we can start to ask and answer questions that 10 years ago, 5 years ago, we just couldn't ask. And it's sort of exponentially increasing, which I think is what's exciting.
As we're getting into wrapping up here, this is a question that I'm asking people this season. When I first initially chatted with you about doing this interview, we were talking about creative thinkers. And you were a little puzzled why I would want to talk to you. But I think there's creativity in all problem solving. So the question I want to ask you is, what does being creative mean to you?
I think science is inherently a very creative process. So a lot of people perhaps who don't work in science don't think of it that way because of their experience in school, where science was probably learning a whole series of facts that you had to, reproduce on an exam paper, or learning a sort of set of rules that you had to somehow apply. But research science is, I think, intrinsically creative, because essentially what you're doing is having to imagine something beyond what's already known. You're having to imagine a process, or, in my scenario, a way that the immune system might be working. You have to imagine beyond what's already known about what happens. So that in itself is inherently creative. I think the distinction between science as a creative field versus the arts, is that in science, what you're creating has to be testable. So whereas if you're writing a book or painting something, you can allow your imagination to run wild, and, it can be as fantastical as you wish. In science, it has to be testable. And I think that's the distinction. But that, to me, is the sort of essence of creativity in science. But then that's the big picture. There's also the smaller moments of creativity. So the problem solving when the assay doesn't work. How am I going to better do this? The looking at the data that was unexpected and allowing yourself to move away from what you thought was going on to what might be going on and what the data's telling you. And also, more and more, it's about how we present the data. That's also creative, I think. How do you explain your work to people? You've seen that I find that challenging sometimes. Also, how do you explain it to patients and the public as well as to other scientists? So there's a creative element to that as well. So I think science is a very creative field. It's just constrained by needing to be testable.
How do you make decisions when you've gone far enough down a road and it's not working? And do you ever go back, like with new data and new things? How do you do that?
I mean, that's, again, a very good question. So there's two difficult decisions, really. One is when you think, this isn't right, my idea was wrong. It's being led by the data and ensuring that your experimental design has been designed well enough so that it really does answer the question. Have you looked at enough samples? Have you powered your experiment enough to be able to believe very strongly in the results? But all assays that you do have their limitations and have their constraints. So you also have to keep that in the back of the mind. The other real difficulty is knowing when to let go of a project and to publish it. That's always really difficult. So when something is actually going well and you're getting lovely data, you can always ask the next question and the next question and the next question. But obviously at some point you have to publish it. And that's really difficult, actually, because there's so many factors that are playing into when you press the button. Some of it's around funding, some of it's around needing to prove productivity and that you're publishing well. Some of it's around anxiety that other people might be gazumping you because you get a feel that someone might be going in the same direction as you, but also making sure that you've got enough data that you can publish well and that it's robust enough and that, that you've done all of the controls and checks and measures that you should do. So that's a real challenge. It's easier, I think, to realize when something isn't working than it is to when to let go of the thing that is.
Yeah, that's fair. Well, Jo, this has been a great conversation, and I really appreciate you giving your time to us today. Dr. Jones.
Thank you for inviting me.
Thank you for listening to Everyone Is. Everyone Is produced and edited by Chris Hawkinson. Executive producer is Aaron Dussault. Music by Doug Infinite. Our logo and graphic design is by Harrison Parker, and I am Jen Coronado. Everyone Is a Slightly Disappointed Productions production, dropping every other Thursday wherever podcasts are available. Make sure to rate and review, and maybe even like and subscribe. Thank you for listening.